Hi, I'm Laurie, and I'll be talking about a feature of human physiology that isn't, strictly speaking, ours. The human body is host to over 100 trillion microorganisms, that live in complex dynamic communities in and on our body and interact intimately with our host physiology, make important contributions to our health. As soon as we're born, we're colonized with these indigenous microbial communities, many of which have co-evolved with our species throughout our own evolution, and therefore have developed those ways of interacting really intimately with our own physiology. They can benefit us in many ways. They can help protect us against colonization by pathogens. They interact with our immune systems to help coach them in which other microorganisms to treat as friends or as foes, and they also can help make vitamins that we can't synthesize ourselves. But the microbiota, which is unique in composition in every person, has also been implicated in a lot of human diseases, ranging from heart disease to obesity, diabetes, malnutrition. And so it's important to understand how we can promote and sustain health-promoting microbiota. In order to do that, we need to understand how these communities change over time, because they are dynamic, and how easily they can get displaced to a sustainable alternative composition, which is their resilience, and how they respond to disturbances. If we think about the human body as an ecosystem, we might be able to apply ecological theory to understanding and studying the human microbiota. And if we borrow a bit from ecology, we can understand the microbiota in kind of terms of a topographical landscape in which each point would represent a different community state, and the community would fluctuate around a central tendency as species enter or leave or change in relative abundance. But the catch is, if that community encounters an ecological disturbance, it'll displace the community out of its fluctuation around the central tendency. If the community is resilient, it'll return back to where it was before. But if the displacement is too big, or if the community is not resilient enough, it could get displaced all the way over into fluctuation around an alternative stable state. The bulk of existing research that's been done on disturbance ecology in the human microbiota, much of which has been done on the gut community, which is where the biggest aggregation of these microbes are. It can contain up to 500 different species, has been done on antibiotics. And it turns out that phenomenon I've just described from ecological theory can happen, that when you take antibiotics, it can be possible, especially if you've taken multiple series of antibiotics, to displace the community into fluctuation around an alternative and, in fact, potentially less healthy, stable state. What I wanted to look into instead was a different disturbance type, which is isoosmotic induced diarrhea, which is as glamorous as it sounds. Those of you over 50 may be intimately familiar with this. It's the same as preparation for a colonoscopy. It essentially involves ingesting a large amount of fluid, which mechanically wipes out the contents of your intestinal tract. And that makes it interesting as a contrast to antibiotics for a couple of reasons. The first is, from an ecological standpoint, it leaves a very different landscape for community recovery than antibiotics do, because antibiotics act chemically and therefore selectively, and they knock out the specific strains that are susceptible to them. But something like a mechanical washout is less discriminate. So rather than presenting a landscape for recovery in which particular strains that were affected have been totally knocked out and some have been largely unaffected, you could get an alternative landscape where everybody is basically reduced in population and you have kind of a race to repopulate the lumen. But it's also interesting for its clinical applications, both out of concern for all of the people around the world who get this procedure as part of their routine colonoscopy screening, but also for our understanding of diarrheal diseases and understanding what the impact of that mechanism might be on host health, not because of whatever agent is causing the diarrheal disease, but because of indirect impacts on the composition of the gut microbiota itself. So to look at this question, what I did over 3 years of research in one of the labs here at Stanford in the microbiology and immunology department, working with David Relman, was to design a study and implement it, which looked at this other disturbance type, iod, I had 5 healthy adult volunteers who took fecal samples over 20 weeks, which basically gives you a snapshot of the gut microbiota. And during those 20 weeks, we used the first 10 to look at baseline dynamics and understand what the stability of the community is before we apply a deliberate perturbation. And then we look at the actual effect of the disturbance itself. And then the last 10 weeks are to look at the recovery process and whether there are any long-term effects. Out of the 244 samples that were generated, we can look at 3 different data types, though I focus on the 16S data in what I'll be talking about here and in my own honors thesis. But the 3 data types are metabolomic data, which is basically the small molecules that are made or modified by these microorganisms. Then after DNA extraction from all the microbes, you can either look at that genetic material wholesale to get an understanding of the total aggregate functional capacity of those microbes, or you can look at the 16S rRNA gene, which you heard about a little bit earlier, which can essentially, in this context, focus or function as a microbial ID tag that can tell you who's there, their relative amounts, and their phylogenetic relatedness, which altogether gives you a taxonomic profile of the community, and you can use that to understand how it's responding. So in total, that study design allowed me to address 3 aims, which were understanding baseline stability. So before we even apply a perturbation, how stable is this community? What's it doing? The second aim is to understand what the actual response of the community is to this type of perturbation. And then the third is to understand long-term effects, or what might be happening in the aftermath. So going into the results, these are all of the samples taken from the 5 participants before the disturbance. So this is looking at the first aim of understanding the stability pre-disturbance. Analyzed with something called Bray-Curtis, which essentially looks at the calculates the distance in the dissimilarity between samples, and then another method, non-metric multidimensional scaling, basically allows us to get a picture of that. So this is not a graph so much as a map in which each dot represents a sample, and 2 dots that are closer together represent samples that are more similar. And what you can see here is that the samples, which are colored by participant, cluster by participant, so each person has a kind of distinctive, unique composition of their microbiota, but that there isn't a lot of change over time. So each person stays pretty similar, and their dots are clustered together. So you do start to see stability over time. Another way of looking at this is by plotting those distances between samples, but now it's pairwise, so all of the samples in one person calculate the distance between each possible pair, and plot the distance in time on the x-axis and the dissimilarity on the y-axis. And what you can see by the fact that as you go farther and farther apart, the samples don't really get much more dissimilar, that also suggests these communities are relatively stable in the absence of an applied perturbation. The second aim was to look at how this community actually responds to this type of disturbance. And this is a different ordination method, double principal coordinate analysis, but the idea with the mapping and the dots is the same. The coloring here is that purple is all samples before the perturbation, red is the first right after the perturbation, and then green is everything after that. And what you can see here, because this analysis was done on all samples from all participants at the same time, you can note that not only are the samples associated with the perturbation all to the outside of the general distribution, but they're all skewed in a similar direction. And so that means not only is there a detectable perturbation, but also there's something common among the participants that's characterizing this response across all of them and driving those samples to be dissimilar in the same type of way. And we can understand what that is by looking at individual types of bacteria within the community. And it turns out that shared feature was a change in the relative abundance of the 2 biggest groups, the 2 biggest phyla of bacteria in this community, which were the Bacteroides and Firmicutes. And as you can see in each participant, right after the perturbation itself, that ratio of Bacteroides to Firmicutes is elevated. I also used a different type of analysis, sparse discriminant analysis, which picks out the most characteristic specific OTUs or species that actually make the samples right after the perturbation different from all the other samples in the participants. And that, again, you can see the pattern in that all the important Firmicutes OTUs are depressed, all the important Bacteroides OTUs are elevated, but this also pulled out a particularly important family within the Firmicutes phylum called Ruminocaucaceae, in which it was a really kind of characteristic among all of the participants after the clean-out that this was a particularly depressed phylum in the response. So I wanted to look into that a little bit further. So I pulled out the shared taxa of Ruminocaucaceae that was most abundant across all of the participants and looked at its specifically abundance over time. And this not only kind of confirmed what the sparse discriminant analysis suggested, that it's depressed after the clean-out, but it also revealed an important other finding, which was even though in those community-wide metrics that I just shown, we saw recovery in approximately 3 days. So those red dots that were associated with the displacement were only the first 3 days. Some of these individual taxa actually take longer than that to recover. So whatever health effects might be associated with those individual taxa, we might not be able to see recovery on the same scale that you see by looking at the community-wide metrics, which suggested that recovery happens around 3 days. So finally, the last question is, there was an observed effect, but what happened on the long term? Well, it looks like mostly nothing, which is probably a good thing. But this is individual, again, ordination of the samples, but now with not only the pre-disturbance samples, but also the post-disturbance samples. And you can see they're basically indistinguishable from each other. The demarcated black circle and triangle are the centroids for each of those pre and post clean-out samples. And you can also see that those are not very dissimilar from one another. So altogether, this suggests that there aren't long-term effects of the clean-out on participants when you look at the taxonomic level at the 16S gene. And again, that's confirmed with an analysis of all of the participants together. So why does this matter? Well, it matters for 2 reasons. The first is more theoretical, because it turns out the patterns that we see in the recovery of this gut community actually echo what you see in traditional ecology research in a creek river system that's been subjected to a flash flood, which is not only kind of cool, but it's also important because it suggests we might be able to pull other things from ecological theory to suggest whose microbiota might be more stable or more resilient. And that's important for us to know if we're going to start kind of engineering medical decisions around the content of your microbiota or actually changing that in order to impact your health. But it's also important, these findings are also important because of their clinical implications. So it suggests those of you getting or who will get those colonoscopy screening preps shouldn't have to worry about the long-term impacts of those on your health through the microbiota, at least. But it's also important as a baseline of comparison for studying the impacts of diarrheal diseases on the microbiota, because the effects that we can see with just the mechanical effect of the diarrhea here, we can contrast with what's seen as long-term impacts on the microbiota with diarrheal diseases, and instead attribute those effects to the inflammation or the direct activity of the pathogens themselves. So the work that I've been presenting here was what I did for my honors thesis I presented last spring, but it has been ongoing with the inclusion of 3 more participants that'll be included before we publish this. And as I mentioned, there's metagenomic data and metabolomic data that we are generating and integrating into this as well. So thank you so much for lending me your ears to listen for a little while. I'd also like to acknowledge my mentors at Stanford, especially Dr. Relman and Dr. Stanley Falco, as well as the sources of funding that I had through Stanford and the other members of the Relman Lab. Thank you.